Hello everyone, welcome to Brain Blitz Audios. Today in this episode of NEET Preparation, we'll be looking at previous year questions of biology in medical entrance examinations in India. Today we're looking at a very important chapter. The name of this chapter is Biotechnology, Principles and Processes. So this is one of the most important chapters when it comes to biotechnology and also the processes that go with it. So this is the 11th chapter in the grade 12 syllabus for biology in CBSE. So today we'll be discussing all of the questions that will be there for this chapter. So let's jump right into our first question. The first question is, restriction endonucleases are used as? So we have four options. Let's look at each one of them. Option A says molecular buildup at nucleotides. This is done by capping enzymes. So it's the function of capping enzymes to undergo molecular buildup at the nucleotides. So option A is incorrect. What about option D? Molecular cement to combine DNA sites. Molecular cement is the function of DNA ligase. So DNA ligase is the enzyme that is used to combine sites of DNA. Option D is incorrect. If you look at option B, molecular degradation to DNA breakup, that is the work of exonucleases, whereas the question here refers to endonucleases. So option B is incorrect. The right answer is option C, molecular knives for cutting DNA at specific sites. So restriction endonucleases, what they do is when you have a sequence, they cut at specific points on a palindromic sequence, which create overhangs. And these overhangs are later used for binding towards new sites. And that's why option C is the correct option. Let's look at the next question. In genetic engineering, which of the following is used? A, plasmid, B, plastid, C, mitochondria, D, endoplasmic reticulum. This question is quite simple, as I would say. Under plasmic reticulum, the function is transport. It's transport of a lot of cellular contents such as proteins, enzymes, fats, etc. So that's the function of ER. So option D is incorrect. Option C, mitochondria, is responsible for energy production. So therefore option C is incorrect. If you look at option B, plastid, most of the plastids like leucoplasts and chromoplasts, for instance, they are involved in storage of food, and then chloroplasts particularly are involved in production of food, that is photosynthesis. So option B is incorrect. The right answer is option A, plasmid. So a plasmid is essentially an extra chromosomal DNA from bacteria, and the function of a plasmid is to act as a vector. So it carries the new gene or DNA sequence to the host organism, and then it ensures that the new gene is combined with the target organism. So that's why option A, plasmid, is the correct option. Let's look at this question. Introduction of foreign gene for improving genotype is called? A, tissue culture, B, vernalization, C, genetic engineering, D, eugenics. So what is eugenics? Option D, eugenics, stands for selective mating of humans to breed out diseases. So this policy and this study of eugenics is deemed as immoral because of the fact that a lot of discrimination and other horrific incidents happen due to eugenics in the name of eugenics. So option D is incorrect. If you look at option C, if you look at option A, tissue culture refers to the growth of tissue, which is a mass of cells, in an artificial medium. So therefore option A, tissue culture, is incorrect. If you look at vernalization is defined as the induction of flowering process in plants, and this is preferably through cold therapy. So what happens is that the flowers bloom after the winter season. So either they put the plants in winter season, or they create artificial coldness, so that induces the flowers to bloom. So option B, vernalization, is also incorrect according to the question. The right answer for the question, introduction of foreign genes for improving genotype, is option C, genetic engineering. So the term genetic engineering refers to introduction of foreign gene for improving the genotype of an organism or a group of organisms. This question is very interesting. You need to find an example of gene therapy among the following. So if you look at option A says production of injectable hepatitis B vaccine. So the hepatitis B vaccine is one of the few rDNA vaccines available. So therefore option A is incorrect because this goes under rDNA technology. If you look at option B, production of vaccines in fruit crops like potatoes, this is an example of vaccine technology. So option B is also incorrect. If you look at option D, production of test tube babies by artificial insemination and implantation of fertilized eggs, this is an instance of using assisted reproductive technologies. So therefore, option D is incorrect here. The right answer is option C. Introduction of gene for adenosine deaminase in persons suffering from severe combined immunodeficiency or SCID is an example of gene therapy. It was the very first instance where gene therapy was used. So option C is the correct option. Let's look at the next question. cDNA probes are copied from messenger RNA molecules with the help of which of these chemicals? Option A, restriction enzymes. Option B, reverse transcriptase. Option C, DNA polymerase. Option D, adenosine deaminase. Adenosine deaminase, that is option D, is an example of an enzyme. And this particular enzyme is used for digesting purines. So like adenine and guanine are digested using adenosine deaminase. So option D is incorrect. If you look at option C, DNA polymerase, well, DNA polymerase is used for replication of a given DNA molecule. Here the question is DNA are copied from messenger RNA, so option C is incorrect. If you look at option A, restriction enzymes, that is also incorrect because these restriction enzymes are used to cut DNA, not produce new DNA from RNA. The right answer is option B, reverse transcriptase. So reverse transcriptase is the enzyme that we use to convert RNA towards DNA. So this is exactly the opposite of the central dogma, which says transcription follows replication. So therefore, reverse transcriptase is the right chemical with which cDNA probes are copied from messenger RNA molecules. Let's look at this question. Electroporation procedure involves one of these four options. If you look at option D, purification of saline water with the help of a membrane system, this is what we call reverse osmosis. So this is reverse osmosis, and we use this in many countries with water scarcity to, use water from the sea by desalination. So option D is not electroporation, so that is incorrect. If you look at option A, fast passage of food through sieve tubes and phloem elements with the help of, electric simulation, the sieve pores are already present, but in electroporation, new pores are formed. So therefore, option A is incorrect. If you look at option B, opening of stomatal pores during night by artificial light, the opening of stomatal pores results in transpiration of water. So therefore, this is an example of induced transpiration. So therefore, option B is also incorrect. The right answer is option C, making transient pores in the cell membrane to induce gene constructs. So electroporation is a process in DNA technology where we make transient pores by heating the cell membrane and then cooling it down in order to introduce genetic particles inside. So that's why option C, electroporation, is involving making transient pores in the cell membrane to introduce gene constructs, becomes the right answer. This is the next question. What is the first step in the southern blotting technique? So southern blotting is a technique that we use in our DNA technology. This is basically to isolate a gene from DNA. So in southern blotting technique, if you look at the following options, option A is denaturation of DNA on this gel for hybridization with a specific probe. Hybridization with a specific probe is known as probe labeling, and this particular step is step 4 in southern blotting technique. We're asking for the first step, so option A is incorrect. If you look at option D, denaturation of DNA from a nucleated cell, such as the one from the scene of a crime, then option D is incorrect because this is used for forensic analysis. So it's a part of forensic analysis. So option D is incorrect. If you look at option B, production of a group of genetically identical cells, this is what we call as cloning, so therefore option B is incorrect. The right answer is option C, digestion of DNA by restriction enzyme. So DNA is a very long molecule, so therefore you have to cut it into small pieces. And after cutting it into small pieces, that is how you can use each DNA fragment and then amplify it in order to conduct the southern blotting technique. Let's look at this question. It's about PCR, very common these days. The polymerase chain reaction, or PCR technology, was first discovered by option A, Kary Mullis, option B, Psyche et al., option C, Craig Venter, option D, Maxim and Gilbert. If you look at option D, Maxim and Gilbert developed a specific DNA sequencing method, which is known as the Maxim-Gilbert DNA sequencing method. So option D is incorrect. If you look at option C, Craig Venter is the person who was behind the Human Genome Project. So he was the master brain behind the Human Genome Project. So his name is associated with the Human Genome Project, but not necessarily polymerase chain reaction. So option C is incorrect. If you look at option B, Psyche et al. So et al means and others. So Psyche, it's basically Randy Psyche. So Randy Psyche, he worked with a group of people, and his contribution is specifically Taq polymerase. So in order to ensure that PCR goes successfully, you need a DNA polymerase that has to withstand high temperatures. So Psyche et al. Was behind the formation of Taq polymerase, which is taken from Thermus aquaticus bacteria. So therefore option B is also incorrect. The right answer is option A, Kary Mullis. So while Psyche et al. Was instrumental in the formation of PCR and Taq polymerase, Kary Mullis was the person who was in charge of developing this unique technology, and the person was awarded the Nobel Prize in Medicine. Basically it's physiology and medicine, so the Nobel Prize for Physiology and Medicine was awarded in 1993 to Kary Mullis for discovering polymerase chain reaction technology. And that has been very useful today as well. Let's look at this question. So this is a question with a mathematical twist. So you need to use a formula in order to solve this question. So over here you see, after 4 PCR cycles, how many DNA molecules are formed from one DNA template molecule? So for this you need to use the general formula. So suppose you have completed n cycles of PCR from one DNA template molecule. You can see that one DNA template molecule it yields 2 raised to n DNA molecules. So over here, n is the number of cycles. So therefore, for 4 cycles, the formula will be 2 raised to 4. So that is equal to 16. So therefore, option C, 16, is the correct option. If you look at option D, 8, that happens when you have 2 raised to 3. It's incorrect. Option B, 32, is 2 raised to 5. So that means 5 cycles. So that's incorrect. Option A, 4, that means 2 square, so it basically means that you have 2 PCR cycles, so option A is incorrect. The right answer for this question is option C, 16, and we use the formula for finding the number of DNA molecules from one DNA template molecule after n cycles of PCR. So that formula is 2 raised to n, and over here, since we have 4 cycles completed, so 2 raised to 4 equals 16 is the correct answer. Now, let's look at this next question. The Human Genome Project, or HGP, is closely associated with the rapid development of a new area in biology called as A, biotechnology, B, bioinformatics, C, biogeography, D, biosciences. So option D, biosciences, is basically biology, so that's incorrect. Option A, biotechnology, is incorrect because it happened so advance in biotechnology were present long before HGP. Of course, it has its contributions to biotechnology, but biotechnology as a subject had formed way before the Human Genome Project even began. Option C, biogeography, is also incorrect. This is basically looking at habitats and evolution, etc. And the right answer is option B, bioinformatics, because one of the most important aspects of the Human Genome Project was to yield or create vast information of biological matter. So it has to be, because you're basically sequencing the entire genome of the human. You're basically, coding and you're basically finding out the code for the entire human genome. That's a molecule that spans over 3 meters. So therefore, to create information with respect to finding out how to do genome sequencing is what made bioinformatics a new area in biology created by the Human Genome Project. So option B, bioinformatics, is the correct option. Now, this question is with respect to matching up the apparatus with the function. So identify the correct match for the given apparatus. So option A is gene gun, and it says that the function is vectorless direct gene transfer. Now, the reason why option A is incorrect is because that it's not vectorless. So the DNA that is injected by a gene gun is actually coated around a metal. So the metal acts as a vector in carrying the DNA inside the plant cell. So therefore option A is incorrect. If you look at option B, column chromatography, separation of chlorophyll pigments. So this is incorrect because the reason is that the separation of chlorophyll pigments was a result of paper chromatography and not column chromatography. Column chromatography is especially used to isolate one single chemical compound from a mixture. So therefore option B is incorrect. If you look at option B, respirometer, it says the function is to find out rate of respiration. The correct answer is a spirometer and not the respirometer. So therefore option D is incorrect because the function is not matching the apparatus. The right answer is option C, stirred tank bioreactor to carry out fermentation process. So basically a bioreactor is is what you call an intense machine that in which we add the initial substrate, and then it undergoes processes such as fermentation in order to mass produce biological compounds such as, say, medicines. So fermentation process is one of them. So a stirred tank bioreactor can be used to carry out fermentation process. That's why option C is the correct option for this question. So let's look at this question. Genes of interest can be selected from a genomic library by using A, restriction enzymes, B, cloning vectors, C, DNA probes, D, genetic targets. So if you look at the following options, you can see that option D, gene targets, is used to refer to homologous recombination done by biotechnologists to modify a particular gene. So therefore, gene target is basically the area where homologous recombination takes place. So option D is incorrect. If you look at option A, restriction enzymes, these enzymes are used to cut DNA, not to select. The question is asking us which of these selects DNA. So option A is incorrect. If you look at option B, cloning vectors are used to clone a DNA fragment. They cannot be used for selecting any gene of interest from the genomic library. These are used to clone fragments so as to add to the genomic library. So option B is incorrect. The right answer is option C, DNA probes. So what does a probe do? It goes and finds out a particular signal. So therefore, in a genomic library, if you want to get a gene of interest, you have to select a gene of interest by using DNA probes. And that's why option C is the correct option. This is a match type question. We need to find out which of these variables are correctly filled. So which of these options give you the correct fill for the variable. So the variable here refers to, parts of the plasmid vector PBR322. So we need to find out what is A, what is B, what is C, and what is D. So if you look at a labeled diagram for the same, you can see that option A refers to ECORI. So ECORI. So if you look at that particular if you look at A being ECOR1 in the 4 options, you can see that option D already becomes the correct option. So this is what you can do. You can use elimination in order to get to the correct answer. And similarly, if you look at B, the corresponding chemical here is BAMH1. If you look at C, it is ampicillin resistant gene, and D stands for the ORI, or origin of replication. So just by finding ECOR1, we found out the correct option for filling up the variables in a plasmid vector, that is PBR322. So option D here is the correct option. Next question, which one of the following palindromic-based sequences in DNA can be easily cut at about the middle by some particular restriction enzyme? So we need to find out which of these options are palindromic. If you look at option A, you see that it starts with C, G, T, T, C, G. But then if you read it from behind, for the second strand from the 5' end, it's A, T, G, G, A, T, A. So as you can see, these are not palindromic, so option A is incorrect. If you look at option B, it's G, A, T, A, T, G. But if you take it from the reverse end, it's A, T, C, A, T, C, which is not palindromic, so option B is incorrect. Let's look at option D. In option D, you can see that CACGTA is on one strand, and on the other strand, it is TGACTC. So as you can see, again, these aren't palindromic, so option D is incorrect. Palindromic basically means reads the same from both forwards and backwards. So the only option that is correct here is option C, where 5' GAATTC goes with 3'. if you look at it from the reverse end, it's GAA TTC, which is the same as that of the obverse strand. So therefore, option C is a palindromic-based sequence, and restriction enzymes work on palindromic-based sequences because they can cut at the same, between the same bases so as to form overhangs, which can be used by DNA ligase to improve. Now, you need to find out which of these statements is not correct about cloning vector. This is the final question for this episode and of this chapter. So which of the following statement is not correct about cloning vectors? So statement A says that ORI, origin of replication, is a sequence responsible for controlling the copy number of linked DNA. Since ORI refers to replication, that means that the number of copies for the linked DNA is partially a responsibility of ORI, or origin of replication. So option A is wrong. Now, remember that the statement is true, but here we're asked to find which of these is not correct, so therefore option A is wrong. If you look at option D, the ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. So ampicillin R and tetracycline R resistant genes are used to ligate alien DNA, so therefore it is true. So since the statement is true, you can say that the option is wrong. If you look at option C, in order to link the alien DNA, the vector needs to have a single recognition site for commonly used restriction enzymes. The statement is true because if you have more than one recognition site, then the DNA would splinter up and become useless. So option C is also wrong because the statement is correct. The right answer is option B, selectable marker, selectively permitting the growth of non-transformants. So what exactly is the role of a selectable marker? The selectable marker is used to select a particular gene from given fragmented DNA. So therefore, from the fragmented DNA, if you want to select a particular gene, you need to use a selectable marker for that gene. So that's why option B is false, because the statement is wrong. The statement is incorrect, so that makes the option correct, because once again, we're looking at which of the following statements is not correct about a cloning vector. And that's why option B is correct. So that's all, folks, from this episode of NEET Preparation. Biotechnology is a very important chapter. We hope you found this episode interesting. For accessing more of our videos on NEET Preparation, you can hit the link in the description down below. You can click that link and be redirected to the playlist. For getting more of our useful and interesting content, don't forget to subscribe to our channel, which is Brain Blitz Audio. So until the next episode, take care, stay safe, bye-bye for now.